Okay, great. So I think I'll go ahead and start briefly with a few introductory statements. I am Carrie Coughlin. I am at Washington University in St. Louis School of Medicine, and I'm co-moderating this session with Dr. Amy Paller at Northwestern. We are very excited for everyone attending our Pediatric Dermatology Research Association session during this SID virtual meeting of 2021. Just to give a layout of where we're going here. So we will have the welcome, our keynote presentation, which we are very excited about. One of the benefits, of course, of these virtual meetings is that we can cross the pond a little bit more easily and be able to interact with folks. So we're very excited to have Professor Sprecher here today. Then we will have a series of abstract presentations and then close out the meeting. We'll try to stay relatively on time and do our best to hit that goal. So I think I'll probably now go ahead and pass the baton to Dr. Paller, who will introduce our keynote speaker.
Thank you so much, Carrie. It's really my pleasure to welcome Professor Eli Sprecher to be our featured speaker today. Professor Sprecher is the Frederick Rice Professor of Dermatology at the Sackler Medical School in Tel Aviv University in Israel. He also chairs the Division of Dermatology and serves as Deputy Director for Research and Development at the Tel Aviv Sourasky Medical Center. He has founded and directed the Center for Translational Genetics when he was at the Technion Institute for Technology. I think most of you know him and certainly have read his papers. He's co-authored more than 300 scientific publications. He's mentored several dozens of students. He's received numerous national and international prizes and honors. His research focuses on the genetic basis of skin disease, understanding pathogenesis, and of course, attempting to translate that new knowledge into innovative therapeutic tools. So he'll speak with us today about Palmoplantar Keratodermas, one step ahead. Welcome, Professor Sprecher.
Thank you very much, Amy, for your very kind introduction. I will try to share my screen now. Okay. So hello, everyone, and it's really truly great to be more or less here with you today. Unfortunately, still virtually, but hoping that we will soon be able to see each other very, very soon. And I'm very grateful indeed to the organizing committee for the opportunity to discuss with you very briefly some recent advances in our understanding and approach to a group of disorders for which our group has had a particular interest over the years, the inherited palmoplantar keratodermas. Things have changed like everything around us quite rapidly in this field as well over the past recent years. And I would like to briefly review with you some of these advances in three contexts: pathogenesis, diagnosis, and therapeutics. So understanding the pathogenesis of disorders as complex as the palmoplantar keratodermas has been instrumental not only in broadening our understanding of the mechanism leading to the pathology of the skin characteristic of these diseases, but as importantly has been critical also for the delineation of the molecular basis of many very basic, very fundamental aspects of the biology of the epidermis, and we will try to give a few short examples illustrating this point. For example, the deciphering of the genetic basis of a substantial number of palmoplantar keratodermas, or PPKs, which are due to defective function of keratins, has led to the characterization, of course, of the very well-known role of keratins in epidermal biology. But it's shown that keratins do not only function as structural proteins, but are also involved in the regulation of many biological functions. In addition, for example, the Naegeli-Franceschetti-Jadassohn syndrome was shown to be due to mutations affecting the head domain of the keratin 14 molecule and its ability to downregulate apoptotic activity in the basal cell layer. So nothing to do with a structural function. Here we have keratins as regulators of apoptotic activity within the epidermis. And similarly, mutations in keratin genes associated with pachyonychia congenita were found to trigger inflammatory signals, which also suggests, of course, a way to alleviate some of these disease-associated symptoms. So again, a non-structural function. And speaking of pachyonychia congenita, recently the study of specific manifestations of this disease has revealed another role for keratin. And I think this is a nice example which really emphasizes the role of the clinician, which come to observe a certain and novel aspect of the clinical manifestation of a disease we thought we knew all about, but we don't. And then through this clinical observation, there is suddenly a novel pathophysiological pathway which is revealed. And this is the example I'd like to discuss with you now. So pachyonychia congenita, we're all very, I guess, aware of the typical feature of this disease, which manifests with nail dystrophy and a very painful palmoplantar keratoderma and oral leukokeratosis and different types of pilosebaceous cyst. And then we have another disease called hidradenitis suppurativa, which, or acne inversa, which is also very well known to all of us, a chronic inflammatory skin disease, which affects hair units of the axillae and groin and the area under the breast. And it is characterized by persistent and recurrent nodules and abscesses, chronic sinuses, scarring, something which looks quite different from the clinical spectrum of manifestation typical of pachyonychia congenita. Now, mutation in 5 different keratin genes have been found to underlie pachyonychia congenita, or PC. And in contrast, mutations in genes encoding a component of the gamma secretase complex have been identified in familial cases of hidradenitis suppurativa, or HS. And these mutations were found to affect notch signaling. Recently, a number of members of the family were referred to us because of a peculiar combination of signs reminiscent of both PC and HS, as you can see here in this picture. And in this specific case, mutation analysis revealed a known variant in the gene encoding keratin 17, establishing a diagnosis of pachyonychia congenita. But interestingly, the patient's keratinocytes demonstrated decreased notch activity, just as typically seen in HS. And transfection of the mutant keratin 17 was also associated with decreased notch activity. And this is nicely explaining maybe the combined occurrence of HS and PC in the same individual. Now, what is quite interesting is that when we surveyed a cohort of more than 800 PC patients, which are registered with the PC project, 25% of these patients reported signs typical of HS, a majority of which were found to be carriers of keratin 17 mutation. So this seems to be actually a recurrent theme, because just as the patient can demonstrate the double phenotype of PC and HS, the group of Regina Betts and later on our group and other groups reported the coexistence of another keratin disorder, Darling-Dego's disease, characterized by these typical axillary pigmentary macules. And and all these groups describe the combination of Darling-Dego's disease and HS in the same patients, in patients carrying keratin 5 mutation. And here too, the patient keratinocytes, which were actually harboring those keratin 5 mutation, were also found to display decreased notch activity. Now, desmosomal cadherins are critical for proper cell-to-cell adhesion, as demonstrated by the consequences of desmoglein 1 deficiency in SAM syndrome, which features a diffuse form of keratoderma as well as acantholysis, as you can see here on histology. However, just as with the keratins, the study of palmoplantar keratodermas has shown that cadherin's role in epidermal biology goes much beyond mechanical adhesion and actually affects a large number of signaling pathways of relevance to epidermal differentiation, as shown, for example, through the study of striate palmoplantar keratoderma. And this was the work of Cathy Green's group. Reduced expression of desmosomal cadherin can, of course, result, on the one hand, from decreased biosynthesis of desmoglein 1, as in striate palmoplantar keratoderma, but can also result from increased proteolytic degradation of the desmosomal cadherin. And this scenario underlies the pathogenesis of a new form of palmoplantar keratoderma that we recently described. Here, affected individuals present with an erythematous diffuse hyperkeratosis of the palms and the soles, as well, and this is important, we'll come back to this in a minute, as well as peripheral peeling extending to the Achilles tendon area and to the anterior part of the leg. So exome sequencing of families affected by this new form of palmoplantar keratoderma, which is inherited in an autosomal recessive fashion, revealed loss of function mutations in a gene called SERPINA12, which encodes a protein called vaspin. And accordingly, epidermal equivalent reconstructed with vaspin-deficient keratinocytes, which have been engineered so as to express very low level of vaspin. So when we use those keratinocytes in order to generate skin equivalent, as you can see here very clearly, those vaspin-deficient skin equivalents demonstrated histopathological features very similar to those shown by the patients, including hyperkeratosis and acanthosis, etc. What is vaspin? So vaspin is a serine protease inhibitor, which efficiently inhibits kallikreins, such as kallikrein 7, which is responsible in the skin for desmoglein 1 and corneodesmosine degradation. And as mentioned previously, desmoglein 1 deficiency is associated with keratoderma, striate keratoderma, but also other morphological forms of keratoderma. And corneodesmosine is associated with peeling of the skin, with the peeling skin syndrome type B. Accordingly, the mutations that we identified in SERPINA12 causing palmoplantar keratoderma were found to be associated with increased kallikrein activity and decreased level of desmoglein 1 and corneodesmosine, which may, of course, explain this kind of double phenotype, the concomitant presence of keratoderma and peeling of the skin in the palmoplantar skin of patients carrying SERPINA12 mutation. Now, clearly, if it is true that this phenotype results from decreased activity of protease inhibitors, then it should also be visible in patients displaying increased activity of the same proteases. So we need to see the same kind of phenotype, whether the protease inhibitors are down or the protease activities of those proteases inhibited by those inhibitors, such as, for example, SERPINA12. And indeed, let's have a look now at this family with a phenotype very much reminiscent of what we just saw in patients carrying mutations in SERPINA12. Those patients were found actually to carry the first mutation identified so far in a gene encoding cathepsin B, which is an epidermal lysosomal protease whose expression has been found to be affected by genomic changes, not point mutation, in another disorder of cornification called erythrokeratolysis emalis. And the mutation in this specific case was found to increase cathepsin B proteolytic activity. And we now have, in fact, a growing number of palmoplantar keratodermas and other disorders of cornification, or other forms of disorders of cornification, including recessive ichthyoses, which result from increased proteolytic activity due to increased protease activity or decreased protease inhibitor activity. It's a kind of recurrent theme. Now, these advances in our understanding of the pathogenesis of palmoplantar keratoderma are not only broadening our understanding of epidermal biology, but are also facilitating the clinical diagnosis of palmoplantar keratoderma, which can be very often quite complex and challenging. As we previously presented on several occasions, inherited palmoplantar keratoderma can be diagnosed using a minimal set of 4 criteria: pathological features, the morphological appearance of the palmoplantar keratoderma, other clinical features, and their mode of inheritance. And the delineation of a novel group of palmoplantar keratoderma that we just described, featuring increased proteolytic activity in the epidermis and sharing the same clinical feature, namely the absence of epidermolytic changes and of extracutaneous feature, the morphology of the palmoplantar keratoderma, and the fact that this keratoderma is water sensitive and inherited in an autosomal recessive fashion and is associated with peeling of the skin. All this demarcates a novel clinico-pathophysiological group of palmoplantar keratoderma caused by increased proteolytic activity and associated with different genes as indicated in this scheme. Now, accurate diagnosis of palmoplantar keratoderma is becoming increasingly important as we are more and more aware of the fact that those disorders are associated with extracutaneous features, some of which may be quite important and even in some cases life-threatening. For example, cancer. So in some cases, the connection between palmoplantar keratoderma and cancer is obvious, and genetic testing is critical to instate a proper surveillance program, just as, for example, as in hereditary ichthyoses, which is associated with esophageal carcinoma, or in PPK with sex reversal, which is associated with squamous cell carcinoma. In some cases, the true value of genetic testing is not entirely clear. For example, punctate palmoplantar keratoderma has been associated with an increased incidence of cancer in one recent study. However, in other series of patients with punctate palmoplantar keratoderma, and in, for example, my own experience, cancer has not been found to be associated with palmoplantar keratoderma. And the question how to handle this issue is still unsettled and is, of course, very problematic when it comes to a situation where you have to counsel a family or a patient with this disorder. And actually, in other cases, genetic testing can even be, I would say, confusing. For example, we have a disorder called Urier syndrome, which features scleroatrophy of the hands and feet. And this is associated with adermatoglyphia, hypoplasia of the nails, and palmoplantar keratoderma. And approximately 15% of those patients will develop a very aggressive form of cutaneous squamous cell carcinoma, usually during the 4th decade of life, and it's usually involving the scleroatrophic skin. Now, the disease was recently found to be caused by mutations in SMARCAD1, which encodes a protein possibly involved in DNA damage repair. But quite surprisingly, our own group discovered a few years ago that mutations in the very same gene, and in one case, the very same mutation, which was later on found to cause Urier syndrome, cause another disorder with no propensity to skin cancer, which is autosomal dominant adermatoglyphia, lack of fingerprints, and that's it. And mutations in the same gene, SMARCAD1, were later found to also cause another disorder, Bassin syndrome, which features the adermatoglyphia in association with facial milia and skin blistering, which are not seen in the 2 other disorders. What is remarkable is that all disease-causing mutations in the 3 disorders affect the splicing of the same skin-specific short isoform of the gene. So I think that there is really no need to emphasize why interpretation of genetic testing in the case of SMARCAT1 can at this stage be quite challenging. Finally, treatment. So progress in terms of treatment of disorders of cornification has not been as spectacular as the progress in the deciphering of the genetic basis of these disorders. But the past years have seen steadily growing numbers of attempts. So one of the most effective form of treatment in the context of inherited palmoplantar keratoderma, in my own experience, is intradermal injection of botulinum toxin, which is unfortunately mostly effective in the context of one disease mostly, and this is Pachyonychia congenita, as initially shown by Swertling and colleagues. Now, in spite of its name, patients affected with PC do not so much complain about their nails as of the sometimes excruciating pain associated with walking or standing on their feet. And many of these patients are confined to, or are at least using, a wheelchair since early childhood. This is, for example, the foot of a young radiology technician, who was a patient of mine who was about really to lose her job. And this is her foot after only 2 injections of botulinum toxin. And needless to say, not only was she back on walking on her feet, she was even able to run and to do all kind of exercise, to go to the gym, things that she could never have done before the treatment. So sometimes this treatment is really spectacularly efficient. Now, botulinum toxin has been shown to reduce the size of subcallosal blisters, and the fact that those blisters are mainly seen in PC and much less in other PPK could explain why botulinum toxin is mostly efficient in PC. We recently summarized our experience with this treatment, and the effect of botulinum toxin on the ability to walk was the most significant one. Now, it is still quite unclear at this stage what is the exact mechanism of action of botulinum toxin in PC. It could mainly affect type C fiber-mediated neural signaling, in addition to its effect on the accumulation of fluid in the blister. It could directly affect neuropathic pain. But what is quite interesting is that it could maybe also directly affect epidermal differentiation, as shown by Maya Fried in our lab, in reconstructed skin equivalents, where it was shown to decrease epidermal thickness and to increase keratin 16 expression. Now, some therapies have been designed to target the mechanism of action of the disease-causing mutation, and I'd like now to briefly discuss at the end of this lecture 3 strategies targeting 3 different modes of action, 3 different type of mutation: dominant negative mutation, loss of function mutation, and gain of function mutation. Now, let's go back to PC and speak about mechanism number 1, dominant negative mutation. So as we mentioned before, PC result from mutation in 1 of 5 keratin genes, which exert what is called the dominant negative effect, which means that the mutant keratin actually interferes with the function of the normal keratin, of the wild type keratin. And the key to treatment here is actually to decrease the level of expression of the mutant keratin. Now, different strategies have been tried here in patients with PC, with some success, including the use of mutation-specific siRNA with many delivery and manufacturing issues. Rapamycin has been used orally with side effects, although it was quite effective, and has been tried topically so far without conclusive results. And statins were initially identified in a large-scale molecular library screen by the group of Irwin McLean, and have now been shown to benefit PC patients in a small number of case reports. So there is some potential here, still not something that we can consider or call a routine treatment. Now, mechanism number 2, loss of function effect. Some mutations interrupt the translation of the mutated gene product. These are the so-called premature termination codon mutation, or PTC mutations. And aminoglycosides have been shown to allow the translational machinery to read through those PTCs, and have therefore been used increasingly over the past few years to treat a number of genetic skin diseases. And these include, of course, epidermolysis bullosa, but also Hailey-Hailey disease, hypotrichosis, and one form of palmoplantar keratoderma, the Nagashima type palmoplantar keratoderma. And finally, mechanism number 3, gain of function effect. Now, Olmsted syndrome is caused by gain of function mutation in the gene encoding TRPV3. Those mutations actually lead to EGFR, to the epidermal growth factor receptor hyperactivation, which in turn lead to keratinocyte proliferation, suggesting the possibility to intervene by using inhibitors of EGFR. And 2 recent papers demonstrated actually dramatic results using this approach, although, of course, one may wonder about the long-term consequences of EGFR systemic inhibition. So this last example, and the many others we have been through quite quickly over the past 25 minutes, nicely illustrate, I think, the fascinating and so rewarding path leading from the deciphering of the pathomechanism of a disease underlying, the pathomechanism underlying a disorder, to precise diagnosis, and at the end of the day, effective targeted therapies. And just in closing, I would like to very much thank the many people who have contributed to studies from our lab that I briefly presented here, and I would like to thank you very much for your kind attention, and once again for giving me the opportunity to be today with you here. Thank you.
Thank you so much, Eli. That was just terrific. What a whirlwind view and very cutting edge. We have a question from Ken Kramer. Ken was asking, I guess, whether you personally have any experience in using antisense RNA for the treatment of PC, or has anyone done that after Sansi Leachman and the group's publication in that very small area so many years ago. Has anyone tried to do anything?
Yes, to the best of my knowledge, this has been the only attempt of intradermal injection. But of course, this is, I think, a very important, even though it's a proof of concept experiment, I think it's a very important, even I would say a milestone in siRNA therapeutics in skin diseases. It has not only, I think, point at the potential, but also at the problems that we have to overcome in order to transform this treatment into something which we'll be able to readily offer to our patients.
Right, I agree. Can I ask you a question about, you early on talked about how keratin is not just structural, but of course has many other roles, and you showed that so beautifully. Many of these disorders have inflammation associated, some don't. Some have a tremendous amount of pain, like the pachyonychia congenita, and other, sometimes often pretty thick palmoplantar keratoderma doesn't really hurt patients. Can you comment at all on what's known about the role of inflammation, the relationship between keratins and other proteins that are in the epidermis in terms of regulating inflammation? We've certainly seen some early suggestions that there is innate immunity that's increased. What do we know about that, and how might that be affecting inflammation and pain?
I think that I like always to try to start from clinical observation. I think that they are always the most informative. And actually, I don't know if this has also been the experience of other of the attendees, but I've encountered from time to time patients who have been misdiagnosed. Patient with EB who have been misdiagnosed as having some kind of autoimmune bullous disease, and actually responded very well to different anti-inflammatory treatments, starting with corticosteroids. And actually, at this very moment, just 10 meters from from my seat, there is a patient which is now in our ward who has a very, very severe form of Cushing disease because he has been given steroids. He's now 32 and he has received steroids for the past 20 years in one of our neighboring countries, and he has actually junctional EB. So, but we know that steroids actually very often will affect positively at least the symptomatology of those patients. So there is, and then if you look at the histology, very often you will see a lot of inflammation. There is a very old paper by my former mentor, Ruben Bergman, showing how eosinophils are very often massively infiltrating the lesions of dystrophic EB patients. And so from there it is, and there are now some data coming from the lab of Pierre Coulomb and also Thomas Magin showing actually how keratins are actually regulating different inflammatory pathways. And there are already some attempts at trying to block some of those pathways. And I know that you have also been involved in some of these research programs. So inflammation is certainly a target. I think that the major question, which is now perhaps the most interesting one, is whether inflammation has anything to do with cell-to-cell disadhesion. So is it just a byproduct of keratin dysfunction and something which the patients are affected with in addition to this mechanical disadhesion, or whether they have, or whether inflammation is actually directly involved in cell-to-cell disadhesion? And there are also data suggesting that inflammation may actually also contribute to cell-to-cell disadhesion, and thereby by targeting cell-to-cell disadhesion, we may actually prevent also, or at least attenuate skin blistering. So this is in very quickly going through many data.
Thank you. Trying to see, I don't see any other questions for you. Does anyone else? Oh, here's one. Can you comment on the role of connexin in the proteolytic pathway, as some patients like Kidd syndrome also frequently may have hidradenitis suppurativa-like features? Is it involved in some of the mechanisms you mentioned?
Not that I know of, but I may have missed that, but I don't know of any connection or overlap between those two pathways.
Okay, fine. Do you want to say a little bit more about the gamma secretase pathway and some of these disorders? Because we've thought about familial forms of hidradenitis suppurativa and the whole gamma secretase pathway, and how does that link? You mentioned Notch, which is certainly integral, but what is the effect on some of these other genes?
Well, first of all, I mean, you know, I didn't give too much detail about the still controversial issue of Notch decreased activity in hidradenitis suppurativa, because this is something which is considered by some as being actually very well funded. So there are actually many data showing that Notch activity is decreased in patients not only carrying mutation in the genes encoding the different component of the gamma secretases, but also in patients not having those mutations, but displaying clinical features of HS. And in our experience, these are actually the majority of the patients. But on the other hand, Notch abnormal activity has been reported in a very, very large number of inflammatory conditions. And the question, which is still not settled, is whether decreased Notch activity, and Notch has been associated with the regulation of the hair follicle formation. So it makes a lot of sense from a biological point of view, but there is no formal proof that this is not a secondary effect of the inflammation which is over there, and not something which is directly connected to the gamma secretase dysfunction. I showed that in the scheme that so there are biological explanation for notch being dysfunctional as a consequence of gamma secretase abnormal activity, but still it might also be indirectly affected by the inflammation resulting from gamma secretase dysfunction, and therefore this is something which is still not completely clear and settled.
Okay, that's great. I don't see any other questions, so I think at this point we'll give you a big round of applause. Of course, you can't hear everyone out there. There's also been something, oh, Janice Schwartz, by the way, just put in a little note here. Janice is from the PC project, talking about Roger Casper, still exploring siRNA treatment, and has had some recent results from extension of Erwin McLean's work, and in some mice, he's doing experiments on them. And she's encouraging people, so please look in chat, to participate in the upcoming PC Project Symposium towards the end of June. So thank you, Janice. And big thanks to Professor Eli Sprecher for being our guest speaker today. And we'll move on now. Carrie, go ahead and introduce our abstract speakers. Thanks so much, Eli.
That was a great presentation. Thank you so much for that. We will dive into our abstracts now. Our first, and Professor Sprecher, if you could unshare, that would be excellent, because then, yes, thank you. Then our next step will be Dr. Tanya Lehky. She is from NIH, is going to be talking with us about the peripheral nervous system in patients with xeroderma pigmentosum.
Thank you, and let me just get ready to share my screen. I take it that's up.
Looks great.
So again, thank you for inviting me to present to the Pediatric Dermatology Research Alliance. I am an adult neurologist, and the first part was totally fascinating to me, since we deal with a lot of genetic disorders, but in different fields. Today I'll be presenting on peripheral neuropathy in xeroderma pigmentosum. And I'm just go briefly in background, since you're more familiar with dermatologic disorders than me, but xeroderma pigmentosum is a rare autosomal recessive disorder with defective DNA nucleotide excision repair pathway abnormalities. It's characterized by photosensitivity and greater than 2,000-fold increased risk of sunlight-induced skin cancer. There are at least 8 complementation groups lettered serially in XP variant. In 25% of XP patients, a progressive neurodegeneration is observed. This is particularly evident in the XPA and XPD complementation groups. There is combined central nervous system and peripheral nervous system degeneration, and that's marked by loss of cognition, cortical and cerebellar degeneration, progressive neurosensory hearing loss, and peripheral neuropathy. In the pictures in the right side, you see a patient with XPA with the skin lesions, deterioration to the point that she is using a walker by age 37. You can notice also she's quite hectic. And then in the bottom MRI images, you see that there is significant cortical atrophy. This is a retrospective study of XP genotype and phenotype patients with nerve conduction studies performed from 1986 to 2015, all at the NIH EMG lab. The nerve conduction studies were compared with hearing studies, brain imaging, deep tending reflexes, and IQ. The deep tending reflexes were used as a marker of the existence of peripheral neuropathy on a clinical exam. There are 106 patients that were seen in Dr. Kramer's clinic for XP genotype phenotype presentations. About half had nerve conduction studies. 21 were excluded because of XP TTD complex, incomplete studies, or other disorders pruritus. Other comorbidities such as cancer chemotherapy. The demographics are that of the 33 patients studied, you will see that the circled in red is the XPA and the XPD complementation groups. These are the only two groups that had abnormal nerve conduction studies. Of note, and that's circled in blue, is that when you look at the XPA, you will notice that there are abnormal sensory nerve conduction studies in both sensory nerve conduction studies and motor nerve conduction studies in 7 of 9 patients, or 78%. And this denotes a sensory motor neuropathy. While in XPD, you will note that there is only 5 out of 10 patients that had abnormal sensory nerve conduction studies, but none of those 5 had abnormal motor nerve conduction studies. And this would be consistent with a sensory nerve neuropathy present in 50% of the patients. You'll also note that there is abnormal hearing, abnormal MRIs, and abnormal IQ in both the XPA and XPD groups. This was not appreciated in the other complementation groups, though the numbers are limited. We did correlation studies with the nerve conduction studies versus hearing, brain atrophy, and full-scale IQ. On the right side are graphs depicting the correlation studies between nerve conduction studies and hearing loss. The correlations between the sural sensory nerve study and hearing loss, MRI severity, and full-scale IQ were significant. In the median sensory nerve, of course, an arm nerve compared to the sural, which is in the leg, they were significant in hearing and MRI severity, but not for IQ. Now for the motor studies, the fibular motor nerve, which is a nerve to the leg, XPA had significant correlations between hearing and MRI severity. This was not appreciated in the median motor nerve because we did not see significant abnormalities in the median nerve. The difference between the fibular, the leg, and median nerve suggests that there is a length-dependent neuropathy involving the motor nerves. For the motor nerves in XPD, there were no significant correlations. There is a limited number of longitudinal studies for the nerve conduction studies. One XPA patient showed marked hearing loss, marked in red, over the years, but only a gradual decrease in the sural sensory response from normal of 10.8 microvolts to abnormal of 3 microvolts over 19 years. The second XPA patient had no evidence of neuropathy yet. In the XPD, again, you saw the marked loss of hearing loss over the years and a gradual decline in the sensory response. Similar findings were seen in 2 other XPD patients, and 1 patient was normal. In conclusion, patients with complementation XPA developed sensory and motor neuropathies, and XPD appeared to develop only sensory neuropathies. This suggests that there may be 2 different mechanistic etiologies involved in the peripheral nerve degeneration. Development of peripheral neuropathies correlate with other markers of neurodegeneration, such as hearing loss, cerebral atrophy, and loss of IQ. A limited number of serial nerve conduction studies show gradual development of the peripheral neuropathy. Hearing loss may precede the abnormal nerve conduction studies, and though we didn't really discuss it, absent deep tendon reflexes appear also to sort of precede the appearance of abnormalities in the nerve conduction studies. And with that, again, thank you, and I'm open to any questions.
Great, thanks so much. I don't see anything in the Q&A, so I'm going to go ahead and ask a question that I had. Thinking about, are you all next working on ferreting out some of these mechanisms? Because I assume, you know, the ultimate goal is there something intervenable to help these patients?
So I know that Ken Kramer is in the audience, so he asked the question earlier, so he may want to say what his group is doing. I think that we did this retrospectively. There's some gaps in sort of our understanding of what all is going on. In XPA, I don't know if you also have motor neurons degenerating, as well as evidence of this length-dependent motor neuropathy. And we also don't know the extent of small fiber etiology, existence. So I think we still don't completely understand it. There've been several papers acknowledging that there's neuropathy in these two groups, but I think this sort of small population but difference sort of tells us that, even though we're seeing a lot of similar central nervous system degeneration between the two groups, it's actually a different thing going on peripherally, subtly, but maybe of significance.
Great, thanks. And I did receive a chat from Dr. Kramer. Sounds like they're looking into whether oxidative damage plays a role into the degeneration in the nervous system. So thank you for that, Dr. Kramer, and thank you, Dr. Lehky. We are right on time. We are going to move to our next presenter. That's going to be Eun Kong. She is from Emory and is going to be talking to us about itch scales in pediatric patients. And your slides look great.
Great, thank you. All right, good afternoon. I'm Eun Kong, a fourth-year medical student at Emory University School of Medicine. Today I'd like to present a comparison of Pruritus assessment tools for children, ItchyQuant, KidsItchyQoL, and TweenItchyQoL. So for our study, we were interested in developing age-specific Pruritus assessment tools for children. We previously developed a quality of life survey and a Pruritus severity scale for Pruritus assessment in adults, namely ItchyQoL and ItchyQuant. In an effort to develop age-specific Pruritus assessment tools for children, we developed novel quality of life assessment tools, TweenItchyQoL for 8 to 17-year-olds, a 35-item survey, and KidsItchyQoL for 6 to 7-year-olds, a 14-item survey. As for ItchyQuant, ItchyQuant is a self-reported itch severity tool that was initially developed and validated in adults and was subsequently adapted and validated for children in both age groups. So in order to develop new items for the TweenItchyQoL, we conducted in-depth open-ended interviews of itchy children ages 8 to 17 until no new concepts were elicited. Transcripts of the interviews were reviewed to identify novel concepts not present in the adult ItchyQoL, the CDL-QI, the PED Quality of Life 4.0, or the Childhood Atopic Dermatitis Impact Scale. For the 6 to 7-year-olds, we asked them to draw a picture of their itch and use the picture to ask them about their itch until no new concepts were elicited. Both surveys contained novel items drawn from these interviews, as well as items adapted from previous instruments, such as the ItchyQoL. The KidsItchyQoL for the 6 to 7-year-olds was cartoon annotated for the benefit of the age group, whereas TweenItchyQoL was not. We assessed age appropriateness of the wording using the Flesch Kincaid readability test. For the TweenItchyQoL, we asked them about how their itchy skin problem has affected them over the last 7 days. For the 6 to 7-year-olds, we asked them both about today as well as last week in an effort to help them focus on their itch and to assist them with differentiating between their symptoms today versus last week. In addition, in these two populations, we validated ItchyQuant, a self-reported pruritus severity tool, with regard to their itch in the last week. In our study, the quality of life survey, KidsItchyQoL or TweenItchyQoL, and ItchyQuant were administered to both populations, 100 children aged 6 to 7 and 175 children aged 8 to 17 with chronic pruritus. Chronic pruritus is defined by duration greater than 6 weeks in any area of the body and with any etiology. All patients were recruited from Emory Dermatology Clinic. All surveys were administered across 3 visits, the baseline visit, a second visit 1 week after the baseline by phone, and a third visit more than 2 weeks after the baseline, either in person or by phone. Both TweenItchyQoL and KidsItchyQoL demonstrated reliability, reproducibility, and validity. As a reference, for the Cronbach's alpha and the interclass correlation coefficient, or the ICC, a value of 0.4 to 0.7 is considered moderate, whereas a value of over 0.7 is considered to be very good. So for both reliability and reproducibility, both tools hit values close to or greater than 0.7. For responsiveness, only the TweenItchyQoL demonstrated statistically significant responsiveness. Though not statistically significant, KidsItchyQoL did demonstrate a strong correlation between the scores and self-reported changes in itch. So from this head-to-head comparison, we hypothesize that the stronger psychometrics in TweenItchyQoL may be due to the larger number of items, with 35 items compared to 14 in the KidsItchyQoL. The lower psychometrics for KidsItchyQoL may be attributable to the fact that 6 to 7-year-olds are a more diverse population in cognition and literacy, with children ranging from kindergartners to 3rd grade. Finally, the ItchyQuant, the severity scale for pruritus, demonstrated responsiveness in both pediatric populations with statistical significance. With regards to reproducibility, since ItchyQuant is a single item instrument, an ICC value over 0.4 is actually very hard to attain and is considered to demonstrate strong reproducibility. In summary, we present 3 promising tools for assessing chronic pruritus in children aged 6 to 7 and 8 to 17, KidsItchyQoL, TweenItchyQoL, and ItchyQuant, that can be used in clinical trials and epidemiological research. The KidsItchyQoL and TweenItchyQoL will allow researchers to gain better insights into the multidimensional nature of quality of life in chronically itchy children. Furthermore, while multi-item quality of life instruments may not be as practical in the clinical setting, the ItchyQuant would be very useful in clinic as a tool to monitor severity of chronic pruritus in children. I'd like to thank my mentor, Dr. Sufi Chen, and all my co-authors who have contributed to this project, as well as our funding. Thank you for your attention, and I'd be happy to take any questions.
Thanks so much for that presentation. I like how you demonstrate the necessity of having the really in-depth qualitative methods before you can actually put together the surveys, and then the kind of rigorous deployment and showing the reproducibility of these and validity with the children. So that's great. We need that in kids. So appreciate that. I don't see other questions, so we'll leave it as a glowing comment. And then move on to our next abstract. Thanks so much, Dr. Kong. Our next presenter will be Justine Seivright. She is going to be talking with us from the University of California at Los Angeles about comorbidities of hidradenitis suppurativa. And your slides look good.
Okay. Hi, everyone. My name is Justine. I'm a medical student at UCLA, currently completing a research fellowship with Dr. Jennifer Shao before starting my 4th year of medical school this summer. Today, I'll be presenting this abstract titled Comorbidities of Pediatric Hidradenitis Suppurativa, a retrospective analysis. The key message of this abstract is that pediatric patients with HS face a high comorbidity burden, including both medical and psychiatric conditions. Further large-scale studies to investigate potentially understudied or underreported pediatric HS comorbidities are needed. Disclosures for all authors and my contact information can be found at the bottom of this slide. The prevalence of pediatric HS in the United States has been reported to be 0.028%. However, the true prevalence may be higher due to underdiagnosis. Though pediatric HS may be less prevalent than reported in adults, HS has a profound negative effect on patients' quality of life in all age groups. Adults with HS are known to have a high comorbidity burden, including cutaneous, cardiovascular, metabolic, endocrine, and psychiatric disorders, among others. However, data on pediatric HS is limited. As such, we aim to characterize the physical and psychosocial burdens of pediatric HS patients. For our methods, an electronic health record search was conducted in April 2020 for HS patients 25 and younger within the University of California, Los Angeles Health System, using ICD-9 and 10 codes for HS. Inclusion criteria included a diagnosis of HS prior to 18 years old. Data on demographics and pediatric comorbidities up until 18 years of age were extracted through chart review. Our study identified 73 patients diagnosed with HS before 18 years of age. 59 were female and 14 were male. Mean age at symptom onset was 12.6 years, with a range from 6 to 17 years. Mean age at HS diagnosis was 14.3 years, with a range from 7 to 17 years. 48% of patients asked had a family history of HS. Overall, comorbid conditions were reported in 93% of patients. Our cohort was racially diverse, as you can see in this pie chart. 36% were white, 28% Hispanic, 19% Black, and 6% Asian. Cutaneous comorbidities reported in our cohort are shown on this slide. The most common cutaneous comorbidity was acne vulgaris, seen in 37% of patients, followed by acanthosis nigricans and atopic dermatitis. Components of the follicular occlusion tetrad, such as pilonidal disease, dissecting cellulitis of the scalp, and acne conglobata were less frequent in our cohort, suggesting that HS could be the first presenting condition of the tetrad. Three percent of our cohort had a diagnosis of precocious puberty, and 2% of our cohort had thyroid disease. Among female patients, 26% had irregular periods, 10% had a diagnosis of PCOS, and 9% had hirsutism. Metabolic conditions reported in our cohort are shown here. Metabolic comorbidities were common. The majority, 52% of patients were obese, which was defined as a BMI above the 85th percentile. Metabolic comorbidities were predominant among male patients. For example, 43% of male patients had hypercholesterolemia, as compared to 10% of female patients. 4% of patients had a diagnosis of hypertension before age 18. We suggest screening for pediatric HS patients to include height and weight measurement, blood pressure measurement, a fasting lipid panel, glycated hemoglobin, and a referral to endocrinology to screen for precocious puberty and PCOS, though expert consensus guidelines for screening are needed. Here are other comorbidities reported in our cohort, including articular, genetic, hematologic, and pulmonary conditions. 8% of our cohort reported arthralgias, 3% had Down syndrome, and 10% of our cohort had anemia. Finally, almost 1/5 of patients had a diagnosis of asthma or other reactive airway disorders. And this data, in addition to the prevalence of atopic dermatitis in this cohort, is notable. There may be a common pathophysiologic pathway explaining atopic conditions in HS patients, possibly related to Th17 axis skew, microbiome dysbiosis, or sex hormones. Investigation of the underlying mechanisms driving co-occurrence of atopic disease in patients with HS is needed. Here are the neuropsychiatric comorbidities of this cohort of pediatric HS patients. Psychiatric comorbidities, including anxiety, depression, ADHD, suicidal ideation, substance use disorder, and bipolar affective disorder, affected 32% of patients. Anxiety and depression were especially common in female patients at 25 and 22% respectively. 3% of patients had a diagnosis of substance use disorder before age 18. And notably, 7% had a history of suicidal ideation, highlighting the importance of mental health screening at HS-related visits. The prevalence of anxiety and depression described in our cohort is higher than in previous pediatric HS cohorts. It reflects the urgent need for physicians who care for pediatric HS patients to perform mental health screening and connect patients with mental health providers when appropriate. In conclusion, HS pediatric patients face a high comorbidity burden, especially with psychiatric conditions. Multidisciplinary care is helpful to care for the patient's HS, as well as their comorbidities. Further large-scale studies are needed to investigate potentially understudied or underreported pediatric HS comorbidities. For example, the relationship between atopic conditions and HS. Early identification, including routine mental health screening and early management of comorbidities, is warranted in the pediatric HS population. Finally, the development of consensus guidelines on comorbidity screening in the pediatric population is needed. Here are my references. And I'd like to give a special thanks to all of our co-authors for their hard work and to Dr. Hoagland, Dr. Shi, and Dr. Shao for their guidance and mentorship. Thank you for your attention.
Thank you for that great presentation. I think you really highlight what we see in clinic with a complex population that needs a lot of services and that has a lot going on, and sometimes visits can even feel overwhelming because you're trying to deal with 4 things in each visit. So really kind of coming up with guidelines, I agree, for thinking about screening and frequency of screening would be great in terms of helping the provider end of things, but remembering to keep in mind to kind of ask open-ended questions and really kind of support this population that has a lot going on is really important. So thanks very much for your presentation.
Absolutely, thank you for having me.
And next we are going to hear from Mary Sun at Yale. She is going to be talking to us about an ichthyosis scoring system.
Hi, everyone. I'm delighted to join you all for the PeDRA session at SID. My name is Mary Sun, and I'm a graduating Yale medical student who's been working with Dr. Stroh and Dr. Paller. And today I'd like to talk to you all about our work on developing and validating a new severity assessment tool for ichthyosis. So here are some photos from a young man who was enrolled in our triferritine clinical trial for ichthyosis. And I picked these photos because I think they do a very good job of answering the question of why it's important to have a validated severity assessment tool. For many patients, ichthyosis is very disfiguring. And because there's no cure, many patients view clinical trials as a source of hope. And without a validated and reliable severity assessment tool, finding appropriate subjects for these clinical trials and quantifying treatment outcomes becomes very challenging. Currently, there are 2 validated ichthyosis severity indices, among others that are currently used in studies, but there's no validated tool to score whole body ichthyosis severity. And the ability to do that is very important because, as you can see here, scale distribution and severity can actually vary quite considerably across the body. So to create a whole body ichthyosis scoring system, we split the body into 10 sites to be scored, including sites of psychosocial significance such as the face. We designed our scoring system to be used in children under 8 and those 8 years and older. And we ask raters to score scale and erythema severity for each body site. Scale and erythema are the 2 most common characteristics shared among the congenital ichthyosis. The scores range from 0 to 4, with 0 being completely normal and 4 being the most severe. Half scores were allowed. So next we needed to create photographic standards for both scale and erythema, representing each of the severity levels. And upon analyzing the thousands of photographs that we had in our National Registry for Ichthyosis and Related Disorders, we noticed that over 95% of patients with ichthyosis fall into 3 broad phenotypic categories. And they include scale predominant, such as lamellar ichthyosis, erythrodermic, such as Netherton's and CIE, and erythrokeratodermic, such as EI. From our registry of photos, we created photographic standards representing all 3 of these cardinal phenotypes. And then we analyzed the photos to come up with written standards to adequately capture the range of phenotypes that you would see for each severity level. So here are some photographic standards for the torso, and this is severity level 1, which is characterized by normal skin intermixed with small scales, as you can appreciate in the first and third image here, or areas of minimally shiny, waxy, thickened skin, like the second image, or partial loss of normal skin markings. The difference between a level 1 and a level 2 is really in the word choice. Level 1, you have normal skin intermixed with small scales. In level 2, you have several areas of scaling upon a background of mildly thickened skin, often with some loss of normal skin markings. In level 3, you no longer have several areas of scaling. Instead, the scales are now confluent throughout the body region, with or without focal areas of thick piled scales and moderately thickened skin. And severity level 4 is characterized by extensive areas of confluent, primarily thick piled scale and severely thickened skin. For erythema, the severity score of 1 is considered mild. It's barely perceptible pink. Severity score of 2 is moderate, it's pink to red. Severity score of 3 is severe, and it's bright red. And a severity score of 4 is considered very severe, and it looks deep red purple. So using the ichthyosis scoring system really boils down to 2 steps. First, you want to score the average scale severity for each body site, and then you score the erythema severity for each body site. And to aid in ease of use, we created free mobile applications available for download on the iOS and Android platforms. All you have to do is enter in your scores for each of the 10 body sites, and it'll automatically compute a whole body ichthyosis severity score for you. So now that we've created the ichthyosis scoring system, our next step is to validate it. And after talking with our biostatisticians, we picked out 9 patients to serve as our test cohort. And these 9 patients provided us with a total of 83 test images to be used. We made sure to represent all 3 of the ichthyosis phenotypic categories, and we balanced age. All of these patients formed the full spectrum of mild, moderate, and severe ichthyosis. Now, to validate the ichthyosis scoring system, you really want to answer 2 main questions. The first is, to what extent do the dermatologists agree in their scoring using our tool? In other words, what's the inter-rater reliability? And that's measured by a statistic called the ICCs, or intra-class correlation coefficients. ICCs range from 0 to 1, with 0 being absolute disagreement and 1 being absolute agreement. Any ICC over 0.7 is considered to be very reliable. And the second question we want to answer is, are the raters consistent with themselves when using this tool? In other words, what's the intra-rater reliability? And that's also measured by ICCs. So a total of 11 adult and pediatric dermatologists participated as raters in our study, and we broke them up into 2 groups. Each group received the same instructions, same high-resolution printed standards and test photographs, and then a month later, we remailed everything to the same dermatologist and asked them to rescore in order to test for intra-rater reliability. And here are the results. The light green bars represent the second cohort, and the dark green bars represent the first cohort. As you can see, for scale, both cohorts achieved excellent inter-rater reliabilities with ICCs of over 0.9. For erythema, both cohorts also achieved very good inter-rater reliabilities with ICCs over 0.85. And then for the total whole body ichthyosis severity score, both cohorts were again in the excellent range with ICCs of over 0.9. This means that there's excellent agreement and consistency among our raters. For inter-rater reliabilities, the results are even more robust. Both cohorts achieved good to excellent intra-rater reliabilities for scale, erythema, and total body severity scores. This means that the raters were consistent with themselves when using our tool. And we have picked the 1-month period because not only would it reduce rater recall of the previous scoring, but it also mimics the routine clinic follow-up timeframe. But while we had substantial intra-rater and inter-rater reliabilities, there are important limitations to note, which is that we validated using only photographs. And originally we had planned to do so in person at the First Family Conference, but that plan was curtailed by the pandemic. And although we included more photographs of ichthyosis patients of skin of color, we still need to include more comprehensively account for all races and ethnicities. Our results were reproducible. Both cohorts achieved near perfect agreement for scale, erythema, and whole body ichthyosis severity scores. And this validates the ISS as a reliable and adequate tool for scoring ichthyosis severity. And with the ease of use, and especially with our app, it's not unlikely that the ISS will one day be globally adopted as the gold standard for a whole body ichthyosis severity assessment. And with that, I would like to thank my mentors, Dr. Cho and Dr. Paller, for providing their expertise and guidance and support on this project. I feel incredibly lucky to have worked with two stars in the field, and they both have really inspired my career path. I also want to thank all the raters who participated in this study, as well as all of you guys here and the organizers of the PeDRA session at SID for giving me the opportunity to share with you our work. Thank you.
Thanks for your presentation, Mary. It's great that you've tried, it looks like you really tried to make it simple, but also really focus on the things that are most apparent and matter and are kind of targetable, again, clinical endpoints when thinking about treatment. So well done. And we don't have any questions, so I think we will move on to our final abstract, and that is going to be presented by Amy Paller, talking about the microbiota of congenital ichthyoses.
Thank you so much. I'm going to go ahead and share my screen. We're going to stay in this theme of ichthyosis. Hopefully everybody can see this. And I'm going to talk with you a little bit about some of the first work ever on the skin microbiome across the spectrum of these forms of congenital ichthyosis. I'm speaking on behalf of a large group of investigators who were involved in this project. Now, I think everybody here knows a bit about ichthyosis. These are genetic disorders of keratinization, and among the orphan forms are some major subtypes. This includes ARCHI, the autosomal recessive congenital ichthyosis subgroup that looks like congenital ichthyosiform erythroderma, as well as lamellar ichthyosis, epidermolytic ichthyosis, and Netherton syndrome, and these all share features of having that epidermal barrier defect, and evidence that we have found of the increase in Th17 pathway skewing in skin and blood. They also have increased risk of certain infections, particularly Staph aureus and fungal infections. So why study the microbiome in the ichthyoses? First of all, we know that it's commonly altered in a variety now of inflammatory skin diseases. We know that skin commensals can modulate innate and adaptive immune responses, including Th17 pathway expression. Many commensals have direct activity against Staph aureus and some against fungi as well. And it's exciting to see topical supplementation with skin commensals starting to come in trials for atopic dermatitis, and particularly those that have been shown to kill Staph aureus. However, there are no studies right now to compare the microbiota among the congenital forms of ichthyoses and with healthy skin. So in this study, rather than using just the ribosomal RNA-16S, we chose to use shotgun metagenomics, which allows the simultaneous capture of the entire microkingdom fingerprint that includes bacterial, fungal, and viral organisms in the same analysis. We swabbed patients, but I should say that one could also do this with tape strips, although a lower yield, extracted the DNA, had library preparation, DNA sequencing, and then read classification. And what I'll be telling you about today is the taxonomic profiling that was found. In this study, we took those patients who were 17 years of age and above with a confirmed ichthyosis subtype. Skin swabs were performed at 3 sites: anterior scalp, upper arm, and upper buttock. Patients were not allowed to shower or use an emollient in the previous 24 hours. At the site, they also could not use any topical medication that could affect the microbiome in the previous week at the site, and could not be on any systemic medication for at least 4 weeks that affected the ichthyosis. We genotyped them, including for filaggrin, to exclude that potential as a modifier, and I will tell you that none of the patients were found to have any of the filaggrin defects. A little bit more about the methods here. The libraries were constructed with barcode adapters. We used paired-end sequencing using the Illumina HiSeq X platform, and we trimmed the reads for quality, and we filtered out for contaminating human reads by aligning them to a human reference genome. And what we found is that that left more than 31% that were usable reads to allow for this unbiased approach. Now here are the demographic and clinical data. We in total had 22 patients with ichthyoses that spanned these various subtypes, and we had 16 controls that were age and sex matched. We also looked at transepidermal water loss on the upper arm and on the buttock. And then we did an assessment of severity with the ichthyosis area and severity index. The scale you just heard about, so beautifully presented by Mary Sun, was not yet available at this time. This is a score that's similar to the EASY and PASI and has erythema and scaling subsets, as shown here. What we found is distinct skin microbiome community structures, showing here the PICO-A plots, looking at Bragg-Curtis dissimilarity species level analysis. This combines the multiple sites. You can see the controls here in blue and the different forms of ichthyosis in a variety of other colors clearly separating out. And when we look at box plots, we can see here again statistically significant differences in this type of analysis. And I will tell you also that when we looked at the individual subgroups of the ichthyosis, each was distinct from controls as well. Using metaphylan 2 analysis, we looked at multi-kingdom abundance of bacteria, eukaryota, that's fungi, and viruses. We found that the relative abundance of bacteria was increased at all of the 3 sites. Fungi overall were reduced, and generally low levels of viruses with some exceptions as shown. We also found that species richness was reduced. That means there were fewer species at each of these sites. And when we look at the lower plots here, you can see that those forms of ichthyosis that were more erythrodermic had the greatest reductions in species richness. We found major shifts in key community members. You can see here the reduction of Cutibacterium acnes and Malassezia globosa, but the increase overall in Staphylococcus and Corynebacterium genera. This was consistently seen with every subtype of ichthyosis as shown here. Now, when we looked across the spectrum of the staphylococcal species, we can see these increases in all of these various subtypes, including in Staph aureus. But it was interesting that the one subtype that really predominated were these groups of Staph caprae and capitis, as shown here. We found increases across the spectrum of the Corynebacterium species, and you can see all of these various subtypes here. And a reduced abundance of the Malassezia, as shown here in all of the subtypes, but particularly we see a little bit more here in lamellar ichthyosis and Netherton syndrome. Now, this wasn't across the board, though, for Malassezia. True for globosa, true for sympodialis. But if we looked at Malassezia sleuthii, and I can't really tell you why, we found that it was actually increased at all body sites, especially CIE and lamellar ichthyosis. Well, what about trichophyton, another organism that's a problem in some of these patients? And we found this detected as sporadically colonizing across the subtypes. We found it in CIE, we found it in lamellar ichthyosis and in epidermolytic ichthyosis, and yet only one of these patients had any kind of a recent history of trichophyton infection. So in summary, the ichthyosis microbiome is significantly altered from healthy skin. Cutibacterium acnes and Malassezia are significantly reduced across all of these body sites. And we think that's consistent with the skin barrier disruption, particularly the lipid depletion, as these like to be at lipid-laden sites. Microbial richness is reduced at all sites, but increases in staphylococcal and Corynebacterial species may reflect an attempt to limit Staphylococcus aureus, especially in this altered microenvironment of ichthyosis. And finally, the skewing that's been discovered towards the Th17 pathway may, at least in part, represent a response to the altered microbial colonization in ichthyosis. And with that, I want to acknowledge my colleagues at Northwestern, colleagues at Mount Sinai who participated, and particularly John Kamen and his team at the Agency for Science, Technology and Research in Singapore. I want to thank our patients, and finally, I want to thank you for your attention, and I'm happy to take any questions you might have.
Thanks for that, Dr. Paller. It's very useful when thinking about patients with ichthyosis, because we want to think about what are going to be their comorbidities, and then again, what are going to be some therapies that could help them, and knowing their microbiota is useful. Interesting to think about some of the fungal possibilities there, and whether there are kind of particular things that we should be recommending to patients based on this knowledge of their bacteria and commensal fungal organisms, or infecting fungal organisms, truthfully.
Yeah, and with all the emphasis that's looking right now at commensals, we've seen more with the, for example, staph commensals, but why not fungal commensals too? And so we need to think about how that might affect our treatment of these patients.
Yep, we do have one question from the chat here from Damien Abreu. He's a medical student actually at WashU right now, going into pediatric dermatology. We're so excited. Saying very interesting work. Has there been any notable correlations between transepidermal water loss scores and then the changes in the microbiomes in these patients?
Yeah, and I see Ken's also asked about the severity and correlations. Yeah. We did these analyses, and I will say that they weren't strong correlations at all. There wasn't really a good correlation with the severity itself. With transepidermal water loss, there was some inverse correlation in some of our patients with lamellar ichthyosis, and we're still trying to figure some of that out, but nothing really that we could speak of in terms of saying, well, those who were drier, for example, had a worse problem with shifts in their microbiome. We didn't see that.
Great. Well, thank you. I will just close out the meeting here then. We had a great variety of presentations, so well done to all who were our speakers today. I'm going to close out with just a couple slides here about the Pediatric Dermatology Research Alliance that has really been growing. We are supporting, cultivating, and building our educators, our researchers, and the research that we're producing. I wanted to put in a plug for the PeDRA researchers who are presenting in the concurrent mini symposia with the structure of the meeting this year. I know people are pulled in a lot of directions, but these mini symposia are available online for a while, and we have presenters in almost all of those sessions. There are also PeDRA researchers in the e-posters, both with the talks as well as the static posters that you can peruse at your leisure. And I wanted to point everyone to our roadmap, as we call it, of pediatric research at the SID. It's available on our website. You can navigate to the meetings portion with the SID link. And this is basically our catalog of all the research relevant to pediatric dermatology that's being presented at the meeting, whether it's from one of our members or not. And the list this year, as we were mentioning before this session started, is quite long, and exciting to see the amount of work that's going on related to our pediatric patients. I wanted to put in one announcement for a PeDRA virtual education seminar that's coming on. It's going to be a series of webinars about alopecia areata. And so there's the link there at the bottom there in terms of learning more and registering. You can also navigate just to that pedraresearch.org and find both the roadmap and this information about these educational sessions. And this is kind of upcoming, but we have links to past webinars as well that have been really educational for our members. And then links here about how to become a PeDRA member, our website, as well as our upcoming meeting. We have decided to go virtual, so that's going to be October 14th to 15th, and we are going to have a stellar lineup of presentations and different targeted sessions there. PeDRA has been wonderful with the granting mechanisms, so people who are interested in research related to pediatric dermatology but need some funds to really get off the ground or keep a project moving, definitely keep us in mind. And then we have more of those educational events listed there. And with that, I will wrap up our PeDRA at SID session for today. I thank you all so much for attending, and I think our presenters, we really had kind of rapid fire science here, and it was great and exciting to think about our next steps for some of these projects. So thank you all so much, and we look forward to seeing you at our annual meeting. Bye, guys.